Good morning, Claire. How are you?
Hey, yeah, I'm good, Paren. How are you?
Not too bad. There seems to be a significant amount of interest in being able to work with difficult targets such as GPCRs and membrane proteins. And that reminded me that you had done some work in this field. So I thought I'd give you a call and pick your brain on it.
All right, yeah. I worked on membrane proteins during my PhD. I was trying to express and purify a membrane protein and then do crystallization trials on it and binding studies. But I was very naive then as to just how difficult all of that was going to be.
Funny that you mentioned that word difficult. That came up a lot during my conversations. Everybody mentioned how difficult and tricky it is to work with membrane proteins.
Mm.
Could you tell me a little bit more about as to why these samples tend to be so difficult to work with?
Yeah, so because they live in the cell membrane, they're hydrophobic and insoluble. They usually have low expression levels, and then
Mhm.
you need to find the right detergents to try and remove them from the membrane. So getting any decent amount of folded and functional protein can be really difficult for membrane proteins.
Oh, wow. You just gave me a great idea in terms of how we can help our customers. Nano DSF would be a great tool for them to be able to track the stability of these proteins throughout the
Yeah,
stages of isolation, purification to actual use in a binding study.
yeah, definitely. Yeah.
Did you happen to do any binding affinity analysis with this as well?
Yeah, we did. So it was mostly the old school radioligand binding experiments, so
Mhm.
on crude cell membranes, but a little bit on purified membrane protein reconstituted into liposomes.
Mm.
But I mean, you might know that radioligand binding experiments, they can be very tedious and you have to use radioactivity. So we were looking for other methods where we could use liposomes or something like that.
Mm. No, I hear you. In terms of radioactivity, that's out. And obviously, SPR came up during our conversation. But then some of the things that they mentioned to me was when working with these really, really fragile proteins, it's sort of hard to incorporate them into some sort of a liposome or a nanodisk, or because of their fragile nature, it's sort of very hard to attach them to some sort of a surface to do
Yeah, what a mess.
bioenrichment. Yeah, what a mess. Yeah. I did talk to them about MST as a very, how do you say, benign technique. I don't know if I'm using the right word, but an easy technique where you could just simply take these hard to work with components and just work in solution without
Yeah.
going through a lot of hoops.
Yeah, I definitely I really do wish that I'd had something like MST or that MST was around when I was doing my PhD because it would have made things a lot easier. Yeah, I could have just looked at it in nanodiscs or liposomes, could have even used the crude cell membranes.
Mm.
So yeah. I mean, I'm almost tempted to try and get some of my little membrane protein and try again.
Let's do that as a sample project. Well, if you want to learn more about how MST can simplify your challenging workflows when working with SPRs or with working with membrane proteins, GPCRs, give us a call or visit our website. And yeah, let's get in touch.
Membrane proteins.
Challenge accepted.
Challenge accepted.